In the following couple of slides, let's talk about how to present a story using one figure. Here's a study to demonstrate a compound, this is called sodium routing, activates microglia to clear amyloid beta. This is the pathology of Alzheimer's disease. When you read this line, it's about a story. It's not just increase and decrease. This is about a conclusion. So the title of this paper is Sodium Routing Ameliorates Alzheimer's Disease-like Pathology by Enhancing Microglia Amyloid Beta Clearance. This is just one picture from this paper published in Neuroscience. How they built up this story. The first set of experiments, they have used the control group. Here is the amyloid beta accumulation Alzheimer's disease model. The treatment group, they have used this sodium routing. What they look at is the relationship between amyloid beta and microglia. They have used the IBA1 as a biomarker to label the microglia. What they have seen here when they do the merge both images, you can see here is how many microglia, which is green, close to your amyloid beta. Here's with the treatment, you have more cells close to the amyloid beta, and this is the control group. Here they even divide into the size of amyloid beta, how many microglia around the amyloid beta, which is the plaque here, in terms of the size of the plaque. So this clearly shows the relationship, but that hasn't given you enough details in terms of clear amyloid beta. Now they have the second set of experiments. Here again, this is the control group without sodium routing treatment, here with the sodium routing treatment. Now they have used a different biomarker to show what's happening there. Basically, they have used the CD68 to show phagosome. So here's your phagosome, CD68. So when you have CD68, which is a green, together with the red, you see the yellow. This is a high power. You see here have much more yellow dots than here. You also have this a lot of this red, which is a microglia here. So what this will show you here is about after the sodium routing treatment, they will have more phagosome production, which means from the intracellular in the phagosome. Furthermore, they have demonstrated the microglia phagocytosis. They use another biomarker, which is FITC A beta 42. As you can see here, this is the yellow one. After you have the treatment, this group again, this is the merge of these two, and to demonstrate you have more yellow dots there, which is really the phagocytosis, the labeling. This is the time effect, even so the time effects. So put these three sets of experiments together. They have showed the more microglia access to the amyloid beta, and you show the phagosomes with the yellow dot, and furthermore you see the phagocytosis. This all come to a single story or conclusion is sodium routine activates microglia to clear amyloid beta. This is a concept. Here's another paper published in the journal called Autophagy, which is a very high impact journal. This is the title of the paper. Now we talk about this particular figure, one figure. All they have done is to use cell line, which is called N9 cells. They try to demonstrate how this compound, borexin, activate this autophagy. First, they have used the time course effect. For example, here they used the ALB, the same concentration at a different time point through the different time points up to 24 hours. Here they demonstrate using Western blot to show the protein expression. By doing that, they calculate the autophagy flux, the process. As you can see here, with the time, they will increase. Therefore, if you only do one time point, you will not be able to show this effect. Then here, what they have used is another compound, which is antibiotics. It's called the bifidobacterium here. Again, they look at the time effects. They see a significant increase in terms of autophagic flux. From here, they choose the 12 hours effect, which is here, and did this experiment. This is the control group. This is the borexin group. As you can see, there are a lot of white dots in it. This is actually called a puncta, which is labeled GFP LC3 in the cell. Actually, they have already showed here, but this time they showed at a single cell resolution. They choose 12 hours based on these results. You can see the logical flow one after another. They have used another control. If this is called a neo control, they have used another control. This is called rapamycin. Rapamycin to activate a TOR pathway induce autophagy. They can demonstrate it here. Actually, this compound is more potent than this one. Then after that, they increased magnification to see exactly where autophagy has happened. Here they have shown using the TEM transmission electron microscopy to observe autophagosomes. You see this mitochondria. Here is the little circle. This is called the autophagosomes. Let's see the autophagosomes using the EM to demonstrate it did happen. So this is about a story. So this is a figure design in a single cell. Use one figure to demonstrate this compound activate autophagy from very basic protein expression to single cell resolution to the high magnification at the TEM level. The third type of figure in the design is trying to fill a knowledge gap. So we know, for example, haloperidol can induce neurite lesion. As you can see here, it's a control. We put haloperidol cause neurite lesion. When we use the propionate, which is a gut microbiometabolite can inhibit such effect. As you can see, this has a reversed lesion effect when we added the propionate. But we don't know what the mechanism behind this. We use the NPY siRNA, RNA NPY to knock down NPY, so this effect, preventative effect, disappeared. So this is to fill the knowledge gap here. We know this is through NPY. Here is another paper published in Neuroinflammation. The title of this paper is about ibrutinib suppresses LPS-induced neuroinflammatory responses in BV2 microglia cells and wild-type mice. With this figure, all they did was to use BV2 cells to demonstrate a mechanism. First, they started with the chronological order of the experiment. As you can see here, they starved the cell by using serum-free medium for 30 minutes. Then they have used this compound. After the compound has been used for 30 minutes, they used the neurotoxin, which is here is called LPS. This is the concentration. After 5.5 hours, they measured the gene expression. So here, it's clearly you will pick it up how they did experiment. This is a combination of a flow chart together with the results. In terms of design, this is 2x2 design on mRNA expression. 2 means this 1, 2, 3, 4. You have 2 factors. This is the LPS. This is the compound. 2 means plus minus, plus minus. Or this minus minus, plus plus. So this is 4 groups. 2 times 2. So clearly you see this LPS treated cells, you have significant increase in terms of inflammation measured by COX-2. And here is the interleukin-1 beta. This be prevented by adding this compound ibrutinib. The same is for here. Other pro-inflammatory cytokines like interleukin-6, TNF-alpha, ENOS, all demonstrate a good preventative effect. This is the GR image. In terms of different cytokines measured, this is control group, GAPDH. So this is a combination of 3. 1, mRNA expression, protein expression using immunohistochemistry, and here to use the ELISA to measure how much in the suspension or the fluid. So all they used is just the BV2 cells, and all use one experimental setup, measured multiple things. Throughout, they have a quantification to go with each measurement. I hope that helps you to design your research figures. Thank you for listening. See you in the next video. Bye for now.